Today we are begin Unit 3 Biochemistry, which is probably one of the most decisive units for CUET PG 2026 Life Sciences paper. If you analyze previous year papers carefully, you will notice that at least 8 to 12 questions directly or indirectly come from metabolism. Enzyme regulation is repeatedly tested. ATP yield questions are extremely common. Fatty acid oxidation and synthesis are favorite areas, and membrane structure is almost guaranteed. So, my sincere advice, do not treat this unit lightly. Here, root memorization will not help much. CUET PG asks concepts based MCQs like why a reaction is irreversible, why PFK1 is regulatory, why NADPH is used in synthesis but NADH in oxidation, why membrane is fluid. We will build all that logics today. Topics covered. First, structure and functions of biomolecules. In that, we will study proteins, DNA, carbohydrates, lipids, and vitamins. Second, bioenergetics. Third, glycolysis. Fourth, TCA cycle. Fifth, electron transport system and ATP synthesis. Sixth, fatty acid oxidation and synthesis. At last, membrane structure and function. Let's begin. Now, first question. Why are we studying biochemistry at postgraduate entrance level? Because biochemistry explains how life actually works at molecular level. When your muscle contracts, ATP is hydrolyzed. When nerve impulse travels, ion channels open. When you digest food, enzymes act. When DNA replicates, nucleotides polymerize. For CUET-PG, very high weightage, pathway-based MCQs, ATP numericals, match the following type, enzyme regulation questions. Also, if you go into biotechnology, molecular biology, genetics, physiology, biomedical research, biochemistry is unavoidable. So, from exam strategy point of view, this unit must be mastered, not just read. Let us begin with proteins. Proteins are polymers of amino acids joined by peptide bonds. Each amino acid has an amino group, a carboxyl group, hydrogen, and a variable R group. There are 20 standard amino acids used in biological systems. The most important concept for CUET PG is the sequence of amino acids determines the final structure and function of the protein. If one amino acid changes, folding can change. If folding changes, function can change. That is the molecular basis of many diseases. For example, sickle cell anemia occurs because of a single amino acid substitution in hemoglobin. So, never underestimate primary structure. Proteins have four hierarchical levels. Primary structure, simply the amino acid sequence. Secondary structure, local folding into alpha helix or beta sheet, stabilized mainly by hydrogen bonds between backbone atoms. Tertiary structure, overall three-dimensional folding of the polypeptide chain, mainly by disulfide bonds, ionic interactions, Hydrophobic forces, quaternary structure, association of multiple polypeptide chains, classic example, hemoglobin. CUET PG repeatedly asks which bonds stabilize alpha helix. The answer is hydrogen bonds. Which structure is destroyed during denaturation first? The answer is tertiary and quaternary. Which level determines active site? Answer is tertiary. Proteins are the workhorses of the cell. They function as enzymes that catalyze reactions. Structural proteins like collagen in connective tissue, keratin in hair. Transport proteins like hemoglobin and albumin. Hormones such as insulin. Antibodies in immune system. Membrane receptors. In MCQs, CUET often gives examples and asks identify the biomolecule. So always classify structural, catalytic, regulatory, and transport. DNA has a famous double helix structure proposed by James Watson and Francis Crick. Two strands run antiparallel, one 5' to 3', the other 3' to 5'. Outside we have sugar phosphate backbone. Inside we have nitrogenous bases. A pairs with T using two hydrogen bonds. G pairs with C using three hydrogen bonds. That is why GC-rich DNA is more stable. Nucleotides are joined by phosphodiester bonds. These details are favorite CUET PG questions. DNA is the molecular blueprint of life. Its functions stores genetic information, replicates during cell division, acts as template for transcription, controls inheritance. If CUET asks which biomolecule acts as genetic material in most organisms, the answer is DNA. Carbohydrates are polyhydroxyalhydrates or ketones. Major examples: glucose, immediate energy; starch, plant storage; glycogen, animal storage; cellulose, structural. For exam angle, which is the most abundant organic molecule on Earth? The answer is cellulose. Functions include energy supply, storage, cell wall formation, recognition molecules like glycoproteins and glycolipids. Important distinction. Plants store energy as starch. Animals store energy as glycogen. Lipids are hydrophobic biomolecules. Includes fatty acids, triglycerides, phospholipids, steroids. Fatty acids are high energy molecules because they are highly reduced. Hence, oxidation of fatty acids gives more ATP than carbohydrates. Lipids function as energy reserve, membrane structure, phospholipid bilayer, steroid hormones, insulation, signaling molecules. Exam type question: which biomolecule forms cell membrane? The answer is phospholipids. Vitamins are organic micronutrients. They are needed in small amounts, but deficiency causes serious diseases. Fat soluble A, D, E, K. Water soluble B complex and C. Many vitamins act as coenzymes. For example, B1, TPP, B6, PLP. These are extremely important for metabolism questions. Bioenergetics is the study of energy changes in living systems. All biochemical reactions obey laws of thermodynamics. ATP is called the energy currency of the cell. CUET often asks meaning of or why ATP is universal energy carrier. Glycolysis is a cytosolic pathway. It does not require oxygen, so it is anaerobic. One glucose gives two pyruvate. Net gain 2 ATP and 2 NADH. Even in absence of mitochondria, glycolysis occurs, important in RBCs. Three irreversible regulatory enzymes: hexokinase, phosphofructokinase 1, pyruvate kinase. PFK1 is inhibited by ATP and citrate, activated by AMP and fructose 2,6-bisphosphate. This regulation is a very high-yield CUET PG topic. TCA cycle occurs in mitochondrial matrix. Each acetyl-CoA gives 3 NADH, 1 FADH2, 1 GTP. It is also called Krebs cycle after Hans Krebs. Functions of TCA cycle. TCA is an amphibolic pathway, means catabolic oxidizes acetyl-CoA. Anabolic provides precursors for amino acids, nucleotides, heme. CUET loves asking meaning of amphibolic. The electron transport system is located on inner mitochondrial membrane, complex 1 to 6. Electrons passes via coenzyme Q and cytochrome C. Oxygen is the final electron acceptor, forming water. ATP synthesis. Chemiosmotic theory proposed by Peter. Proton gradient drives ATP synthase. This is oxidative phosphorylation. Very frequently asked. Name of the scientist: beta-oxidation occurs in mitochondria. Each cycle shortens fatty acid by 2 carbons, produces acetyl-CoA, NADH, FADH2, very high ATP yield. Fatty acid synthesis occurs in cytosol, requires NADPH, acetyl-CoA, manolic-CoA. Fatty acid synthase complex catalysis reactions. Oxidation and synthesis are not reverse of each other. Fluid mosaic model. Cell membrane model proposed by Singer and Nicholson. Phospholipid bilayer with proteins folding. Dynamic fluid Asymmetric. Functions of cell membrane: selective permeability, transport, cell signaling, cell recognition, structural support. Quick revision. In this video, we have revised protein levels, vitamin coenzymes, PFK1 regulation, TCA outputs, ETS complexes, chemiosmotic theory, beta-oxidation versus synthesis, fluid mosaic model. Alright students, this slide shows the student support package which is optional but very helpful for CUET PG 2026 Life Science preparation. It includes Unit 3 detailed PDF notes, previous year based MCQs, most expected questions, personalized study schedule, quick revision sheets, and practice sets. These materials are designed each according to CUET PG pattern so that you can revise faster and practice more efficiently. Remember, material alone will not clear the exam. Your daily revision and MCQ practice matter most. These lectures on the channel are provided completely free for life science aspirants. If you find the session useful, do subscribe and share with your friends who are preparing for CUET PG.